Welcome. These lectures are being recorded for RCBC general biology students, but we hope others will join in. We're continuing our discussion of biotechnology, this really amazing field which is impacting so many different disciplines, so many different industries. And we have talked specifically about how it's impacting medicine, how we are creating medications to treat diseases like diabetes through recombinant DNA technology. And we've begun a discussion on how we are using biotechnology to diagnose diseases. In the previous lecture, we talked about Huntington's disease and using electrophoresis to be able to diagnose whether somebody has the gene for Huntington's even before they develop any effects from that gene. Now we're going to move on to expanding that concept to something much larger. And this is a gene chip technology or a microarray. And in this case, this is Affymetrix gene chip. We are evaluating thousands of genes at one time. On this chip within this chip is a microscope slide. And in that slide, we have a million different sort of dots or spots. And on the slide, DNA is sticking up out of the slide. This might be a piece of DNA. It's single-stranded, although it doesn't indicate that on the slide right now. This may indicate DNA that is complementary to BRCA1. This might be another piece of DNA that is going to match with the sickle cell anemia gene. This may be another piece of DNA that's going to match Huntington's. Right? So we have all these pieces of DNA sticking up, and each one of them has a sequence which will match with a particular gene. When the patient's DNA or sometimes RNA is then added, it will complementary base pair with the probe that was on that microscope slide. And they will light up in different colors depending on whether the match was made or not. Did hybridization occur or not? Did complementary base pairing occur or not? So as I said, we are able to identify thousands of genes in one test. And we can do a couple of different things. We might be able to see which genes are present. And then the next step could be which genes are turned on, right? Maybe the gene is there, but it's not making mRNA. It's not being transcribed. So we would say the gene's not being expressed. Okay, now what would that look like? We would take messenger RNA from a patient's cell. We would then create cDNA with it by using reverse transcriptase. And we're going to use fluorescently labeled nucleotides so that the patient's cDNA now is going to fluoresce. And we're going then to add it to the microarray. And remember, on this microarray, we have single-stranded sequences that are sticking up and out of there that may match the sequences that we see in the messenger RNA. So we then would go to, after we added the sample, we would put the microarray in a fluorescent microscope. And then as you can see here, if there was a match, we would see, in this case, we would see green. So where we see green here means that there was a match of the patient's cDNA with the DNA that is on the microarray. The colors may differ depending on the assay that you're looking at. So what I'd like to do is talk to you about the potential of that. Could you walk into a doctor's office one day and 1,000 different genes are evaluated in a few minutes? Maybe, but that's not where we are right now. Where we are right now is evaluating diseases, maybe large umbrella type of diseases. And in this one here, I will try to simplify it, but this is a microarray that is done to evaluate leukemia patients. And we want to not only see what medication might work for them, but whether they're likely to relapse or not relapse. And but I'm going to talk about it maybe even more generically, and just look at it as cancer patients. And what I want you to just do here is take a look at this one lane. This is not electrophoresis, but it looks like it here. So this is one individual patient. This really bright green is one particular gene that we have identified that's related to cancer. This is another gene, the darker one, and then another and another. So if we counted all of these, what are there? Maybe 100 different genes. So we create a microarray and we put the sequences, single-stranded sequences for those 100 different genes that we know are related to different cancers. Remember, we know that all cancers have a problem with DNA. And we've said that instead of evaluating whether something is breast cancer and giving you a certain treatment or prostate and giving you another treatment, what if we looked at the mutation that you had and then provided a treatment based on the mutation, not where the cell exists in your body? In this case, what I want you to imagine is that this is probably the first 4 or 5 people. These are 5 individuals here who, if we look at which genes have been turned on, we can see that genes 50 through 60 are lighting up. In this case, this is a positive. There has been a match between the patient's RNA and the DNA that's on the chip. And you can see that these 10 genes lit up. But interestingly, when we look at the next 10 patients, we don't see those genes lighting up at all. They actually have a different set of genes, or a different set of genes that are turned on than this first group. Now, of course, we didn't just get lucky and have the 4 people that matched all be in the first 4 lanes and then the next 10 that matched. What we do is we use computers to sort these for us so that we can see whole groups and start to understand how individuals within that group will maybe respond to a drug. Now, you can see this next group of, say, 10 people, they turn on a different set of genes, not the one that was expressed here and not the one that was expressed here. And then if we jump to where you see this red here, again, this group turns on a whole different set of genes. What do we see here with these individuals? They're not showing any cells lighting up. And so there are other genes that are related to this type of cancer, which we have yet to identify. Now, what is the value in this? There's tons and tons of value, but I want you to think about it this way. If you have a medication that treats this mutation, or it treats cancers with this mutation, if you know that the patient has that type of mutation at the onset, at diagnosis, then you can provide the medication that is needed for those individuals. If you give that same medication to these individuals, you may not see a response in them at all because the medication may be directed towards this gene mutation, not this one. And then there should be yet another medication for these individuals. Every second you get to get a jump on the cancer, to give the right medication to these patients, you have a better chance of beating that cancer, right? Early diagnostics is key, and that's why we have something that we call preventative medicine. Maybe not preventing it, but early detection. And so if we know the medication that matches a mutation, and we can figure out what mutation a patient has, we can give them hopefully the correct medication from the onset. This is the power of a DNA microarray. There's much more in the future that we can learn from this, but this is the evaluation of thousands of genes within one test. I hope this helps. Stay safe and take care.